Right, good morning everyone. So, antibiotic resistance 2, the first half of which will tally with the cell wall from last time, because we'll study resistance to vancomycins. And then the second part will be about a completely distinct target, the fluoroquinolones. There's some awesome science in this 50 minutes, which I'll document now. So the first thing we're going to look at is vancomycin, our drug of last resort. So it's the best characterized member of the glycopeptide family. And I've put two other members there, but they're much less well characterized than vancomycin. And some of these work in slightly different ways, but we'll concentrate on this. And it was isolated, like most of our other decent antibiotics, from a soil bacterium way back in 1958. And it's our drug of last resort that's only active against gram-positives because this structure is too big to get through the porins in the gram-negative outer membrane. And it's chiefly administered through IV use. And there's other caveats to that. It has some mild toxicity issues. And you have to really monitor the patient whilst you're dosing them with it. So it really is our drug of last resort in that sense. And it's made by NRPS machinery, so this is non-ribosomal peptide synthesis. And what I've done in that figure is try to give you an appreciation that it is initially made as a polypeptide. So where these red numbers are, you can see each individual amino acids. So there's one there with a side chain. Look, that's a modified leucine. There's two, which is a modified tyrosine. Another peptide bond, 3, 4, 5, 6, 7. So even though it looks completely wacky, it starts out life as a 7 amino acid peptide. But then look at all these modifications you get. 3 of the aromatic rings get covalently fused to one another. You have this disaccharide attached. And you even have chlorination of it. So it's a really specialized entity. It's a real specialized chemical entity. So put it all together, we've got rings that are coupled. We've got amino acids that are not standard amino acids. We've got amino acids that have chlorine in them. And the target for vancomycin, it's really cool, is the dealanine termini of the wall. But it's completely distinct from beta-lactams. Now I'll show you why in a minute. But it has the same functional blockage of transpeptidation and penicillin binding proteins because they can't get to their substrate. So instead of directly inhibiting these enzymes via their active site like beta-lactams do, vancomycin instead binds like a cage over the substrate, and it prevents the substrate from being used by the enzymes. So it's two very different ways of working. But in common with beta-lactams, you'll end up with no cross-links in your peptidoglycan, and you'll lyse the cells. And vancomycin is a very powerful drug in this sense. This is a cool statistic, right? So in the first 20 years of vancomycin use, there was no clinical resistance. You could use this and you could be sure it was going to work. And then you say, all right, so what went wrong? And lots went wrong. And it's one of the first pointers where we're misusing the drugs. And it's the misuse of the drugs that drives resistance. So I've put down here the dangers of agricultural use. So this is global, but I've decided to choose the Netherlands. It's a really good case study for this. And look at the statistics. They were using 1.5 kilos per year of vancomycin in a clinical setting. They were using much more, 80,000 kilos of an analogue, avoparcin, which has the same activity, for animal use. So this Now that we know more about antibiotic resistance, this is a really stupid thing to do. You do not use your drug of last resort in animal husbandry. You just don't do it. You keep it for clinical use against humans. So where, you have to ask the question, where's the evidence that links misuse in the agricultural setting with resistance in the clinical setting? So these are these 4 pointers here. The first thing you see is that vancomycin-resistant enterococci, VRE, were found in animals that have been fed the analog avoparcin. So this is a correlation between the avoparcin use and vancomycin resistance. Now the second thing you saw is the vancomycin-resistant enterococci were more common in geographic areas where this avoparcin was used. So that's the second element of linkage. It gets stronger and stronger as we come down this list. The third thing you could say is that if you're testing for VRE in food, it was more common in avoparcin-fed animals. So by now, all the alarm bells are ringing. And then if you tally points 1, 2, 3, you find that the vancomycin-resistant bugs were more common in communities that ate the food that was produced from avoparcin-fed animals. So there's this distinct line of evidence now that's linking this misuse in an agricultural setting with resistance in a clinical setting. And I could give you strong examples of this, but this is kind of a hilarious example of this because the man survives. I can use the word hilarious. So this is an example where a forklift truck driver who worked in this agricultural setting crashed his forklift truck, and here we go, compound fracture of his leg, and he worked in this environment where they were using avoparcin in the animals, and he got a vancomycin resistant infection in his bloodstream because of this. So this is a real positive link between the two types of resistance. The other thing you see is that as our drug of last resort, we do not want to see cross-resistance with other resistant bugs. And this is the first alarm bells that's happening in 99, where we see vancomycin resistant in a patient that has acquired an MRSA infection. So we do not want co-resistance against lactams and vancomycin. We'll be in a big trouble if this becomes clinically prevalent, which it hasn't at the moment, but this is just an example that is possible. So when we're talking about resistance to vancomycin and related compounds, I've just put this note on the bottom. Intermediate resistance is not full resistance. Intermediate resistance is when a bacterium, which normally has a cell wall of a certain thickness, and this is susceptible, it can become VISA, vancomycin intermediate susceptible, just by making a thicker wall. Because what you're doing there is you're using, if vancomycin binds the cell wall substrates, you're just titrating it out. You're just using the wall and more of it to sort of suck the effective concentration of vancomycin out. And this is only intermediate, because obviously you could increase the concentration of vancomycin and still kill these bugs. So this is not true resistance, but I mention it here in this context, right? So we know it's very dangerous that bugs can become resistant to this drug of last resort. How do they do it? And it's really neat. It's acquired resistance of an altered target in the biosynthetic sense. So if we go back to our beta-lactam lectures, we've got our PBPs, and we can mutate them to an alternative form that will no longer react with beta-lactams. That gives us beta-lactam resistance. And this is altered target in a mutational sense. You've just mutated the target. But what we have here with vancomycin resistance, because it binds the substrate for the enzymes, we have to make a different substrate if we want to be resistant. And it was observed in the mid-80s to the 90s, and there's these two cassettes. Now these are two totally different types of resistance. You can either have Van-ABD resistance or Van-CG resistance, and what they do is they turn the diala-diala dipeptide of the cell wall into a different entity, so vancomycin won't bind. So the first cassette, ABD, turns diala-diala into diala-D-lactate, and this is the most powerful means of resistance. This subtle change gives you 1,000-fold loss in vancomycin affinity, which is massive. The second one, diala-diala gets changed to diala-D-serine, and this is less effective but will still give you resistance. And this is a 6-fold loss. So all you're doing, here's your cell wall backbone with your appended 1, 2, 3, 4, 5 peptide arms. All you're doing is you're switching out that 5th position for a different entity. And if you look at the strongest form of resistance, this is the regular substrate, diala-diala, and when you switch to diala-D-lac, it is isosteric, has the same shape, and what you've done is removed the NH of the peptide bond and replaced it with an oxygen. And in doing so, you have 1,000-fold loss of affinity to vancomycin. The resistance operon contained in these genes, this is a transposable element, TN1546, and they're organized. You have two genes, VAN-A, VAN-S, that are regulatory. So these sense the presence of vancomycin and turn on the other genes. You have 3 genes that are responsible for the chemical nature of this switching, and I'll go over them in a moment. And you have 2 genes that are accessory that help resistance in other ways, and I'll describe those as well. So this is the normal pathway of cell wall biosynthesis that we know from lecture 1. You take L-alanine, you do a racemic conversion to turn it into D-alanine. You have a ligase that puts 2 together to give you the dipeptide, and then the dipeptide via mu-ref finds its way onto the tripeptide to make the pentapeptide. And this process now has to be subverted to give you a resistant bacteria. So how does it subvert it? It's really quite clever. This is a belt and braces approach. The first thing you need to do is, if you're making a normal wall, you're going to get killed. So the first thing you have to do in the presence of vancomycin is remove that normal substrate. So this accessory gene, VAN-Y, is a hydrolase that will take the pentapeptide and clip off the D-alanine to give you a tetrapeptide. And so you've removed the target of vancomycin. VAN-X will also help you out by chewing up the diala-diala dipeptide intermediate. And this is the second gene in here. But you still have to build a wall. So what you do is you channel pyruvate using VAN-H into lactate. Now VAN-A is behaving like your ligase, but instead of sticking 2 molecules of D-alanine together, it takes one and sticks it to D-lac to give you this pseudo dipeptide, although it's not a peptide linkage. And then this is a good enough substrate for the regular mu-ref to be incorporated into the cell wall. Depsipeptide It's just, instead of having that regular peptide linkage, it's an oxygen instead. So it's just a peptide where the NH has been replaced by an oxygen because we're using lac instead of an amino acid. So it's really cool, right?
So the transposable element that encodes all this information, was that just selected for when vancomycin was used, or did they just come up with
It is selected for, and it's a product of the fact this is a natural compound. So in the environment, you have bugs that have to be resistant to this. And it's heavily selected for if you're using intravenous vancomycin to treat a patient. But of course, they have to acquire the resistant bug, not the susceptible one. So you've got these two pathways that edit out the existing wall and make a new one. And throughout this lecture, I give you the little PDB codes here so you can do a comparison and look at them yourself. The first thing you have to do is select out dealanine by using this different Van A ligase. So this is your regular deala-deala ligase. And you've got residues. Don't worry too much about the complexity of it. This will be your ATP. And you've got residues that next to your ATP. So the reaction is you activate one dealanine using the ATP, and then a second one comes in and breaks that bond and makes deala-deala, and we lose phosphate. So what we have to do is we've got our ATP binding pocket and our two substrates. And in the regular ligase, in the regular wall, in a susceptible organism, you need to bind these two substrates. And you've got a tyrosine here that allows the first deala to bind. You've got a serine up here where the second one binds. And what you see is in our regular ligase, that tyrosine activates the NH to attack the second dealanine, and you'll make a dipeptide. But in the resistant form, you don't want to make a dipeptide, you want to use lactate, and in doing so, effectively what you do is you switch that tyrosine for a histidine, and that allows activation not of an amine, but of an oxygen, which will react with the deala, and then that will give you that deala lactate depsipeptide instead. So Van A is your sort of first port of call into making the unusual substrate that allows you to be resistant. This is a really nice structure. This is vancomycin in complex with a small peptide that represents the cell wall. So this peptide is in yellow, vancomycin is in green, and it's just glorious. What you see is those three fused rings make a cap, and that cap comes over the fifth position alanine here. Here's the fourth position, here's the third, but essentially vancomycin is just a cage for positions 4 and 5. And the regular sense, when you've got deala with a peptide bond between them, this oxygen of vancomycin makes a hydrogen bond with the NH of the peptide linkage. Now this, if we're having a resistant substrate, would be lactate, and that would be an oxygen, and it would clash, and it doesn't make a hydrogen bond. Okay, so in the susceptible form, the peptide makes the hydrogen bond. In the resistance form, there's a clash because that NH has switched for an oxygen. And it's amazing, right? Vancomycin resistance comes from the alteration of one hydrogen bond, which is remarkable. So, alteration of just one tiny interaction will give you resistance because the clash means it won't bind. Now remember, there's a second type of resistance where you have deala-deserine instead of deala-delac, and this is weaker. This is only a 6-fold change in affinity for vancomycin. And the obvious thing you can say is the difference between serine and alanine means you'll have an extra OH here, and that will clash with the aromatic cage of vancomycin. So this is how, it's nothing about hydrogen bonds because you've still got the NH, it's about the steric clash inside the cage. And the cassette for changing diallo-diallo to diallo-D-serine is almost identical to the cassette described for lactate. So you have an editing enzyme that removes the regular substrate. You have a hydrolase that turns over the dipeptide and channels substrates this way. You have a racemase that can turn L-serine into D-serine. You have a ligase that couples it, and then MURF puts it into the regular cell wall. So the two modes of resistance are probable examples of divergent evolution. Now, if you take the argument that resistance comes about from the switching of a hydrogen bond interaction, you should be able to make a vancomycin that can tackle that, right? If you know the molecular details of resistance, you should be able to make a, but look at this, right? Look at this compound. If you take the best chemists in the world and you ask them to make it, they would probably firstly punch you in the face. And secondly, they would just be overawed by the sheer amount of chiral centers, linkages. But somebody did this, right? This is remarkable. Somebody made a synthetic vancomycin from pure chemistry. Okay, so you've got the normal case where vancomycin here has this oxygen that selects for the peptide linkage. And what you could do is you can manipulate this so if it binds the diallo-D-lac, you could make that a nitrogen in this case, and then you restore the hydrogen bond interaction, right? So in the regular case, you've got the diallo-diallo peptide linkage, making a very nice hydrogen bond to vancomycin. But in the resistant form, where you've switched it up from an oxygen, what you could do is make a modified vancomycin that turns the tables. And somebody did this, and it's amazing, and it works, right? You can take this modified vancomycin, where you've made probably like a nanogram of it, and you can kill the resistant bacteria. But the sensible question, okay, is what's going to happen then when I add my switched vancomycin to resistant bacteria? What do you think is going to happen? They just make the normal wall, right? And it'll clash with that. So this is a very difficult thing to do. And what you have to do is remove the interaction entirely by just generating a species with no hydrogen bonding potential. This is a neutral vancomycin, and then it will kill both the resistant strain and the normal strain. But there's no way we can make enough of this to be clinically. This is just an academic exercise. There's no way we can make enough of this to be clinically useful. The other wacky thing that people are doing in the field, and this kind of blows my mind a bit as well, is they're making a hybrid with two warheads. The first part of the drug is vancomycin, and the second part, they chemically attach a beta-lactam to the top. So the vancomycin goes in, binds the cell wall, stops it from being utilized as a substrate for transpeptidases. The second part of the molecule, when the transpeptidases come along, makes a covalent linkage via the beta-lactam ring. So this is like a double warhead. But again, this is an academic exercise. This is too complicated for clinical use. Cool, they could do it though. Right. Now, most methods of resistance I tell you about are acquired pathways where you do something new, or mutational pathways where you mutate something existing and make it a target no longer. This is a weird example where you can get resistance from altering a signaling pathway. So this is the vancomycin analog with the catchy name, DSA A47934. Behaves just like vancomycin. You add it to the bugs, the sensor will pick up on this and trigger the resistance enzymes downstream in that resistance operon. But what we can do is discover bugs making a variant of this where they've sulfated one of those fused rings. So here's a sulfate group here. And the neat thing about this is the bugs are resistant. And they're not resistant because of interaction with substrate. They're resistant because the sulfated analog doesn't trigger the Van-R sensors. So they never turn on resistance. This would be a pathetic thing to use clinically, because the only thing you would see is if you use this clinically, you get a mutation in the regulators that makes them permanently turned on. Okay, this is how you would then get resistance against this. But this is an interesting idea, that you can reduce the inducible nature of a resistant gene cassette. Right, part 2. Quinolone antibiotics. They're really neat. Massive global usage of these. So, what you see here is they're most commonly used in urinary tract infections, UTIs. Oh, good Lord, what an old reference, 1997. But even pre-millennium bug, $3 billion worldwide sales. And they're a neat example of entirely synthetic antibiotics. So they were discovered way back in '62, and the father of the class is nalidixic acid. And what they were looking to do was make quinine analogues against malaria. But they discovered that this quinolone variant wasn't very good against malaria, but was very good at killing bacteria. So it's an entirely synthetic class. And in the 1980s, we modified this class further by introduction of a fluorine atom. So here's the parent compound, nalidixic acid. Now we talk about fluoroquinolones, where at the C6 position, we've modified it with the fluorine, and I'll tell you why that makes it better molecularly. But they have a good spectrum against gram-positives and gram-negatives, and their target is DNA topology. So when you're a bacteria with a circular chromosome, and you decide to reproduce your chromosome, you've got your replication fork, and you unwind the existing DNA, you replicate. I won't bother drawing the Okazaki fragments. And you replicate, and you end up with one new copy in the existing copy, and you discover, because of the nature of the process, the genomes are catenated. So like a set of magician's rings, they're fused and they can't be physically separated. And the target of these drugs is the machinery that physically separates the daughter genomes after reproduction. And it's amazing. It's two members to this family. The first is DNA gyrase, and it performs this task. And it also will relax these supercoils that exist in front of the machinery. So you can imagine the DNA much like, okay, you've got mobile phones, but old school telephones where it would entangle, and you have to relax that if you want the replication machinery to proceed. So these supercoils are removed by DNA gyrase, and topoisomerase is more responsible for this decatenation. And here's a picture of entangled chromosomes, so you can get the idea that physically this is a very difficult process to catalyze. If you've got two co-joined circular chromosomes like that, you have to open one up, physically push the other through, and then close the second. And if you have any interference in this process, you either end up with broken DNA and a dead organism, or you end up with them still co-joined, the chromosomes, and you can't share them between the, you can't do cell division. You can't effectively separate the genomes from one another. So it's a very powerful means of inhibiting bacterial viability. So how do they work? Right. So class 1 topology control enzymes we're not going to deal with at all. So class 1 are less interesting. What they do is you've got two strands, they cut one of the DNA strands, and they allow the other one to twist, and then they reseal the nick. Class 2 are much more interesting ones, where you cut both strands, allow them to relax, and then put the strands back together again. So the easy way to think about this is class 2 effectively pass one segment of DNA through another. We can study this in yeast, and these class 2 enzymes are present on a single polypeptide that dimerizes, or we can study these in bacteria where they're present on two separate polypeptides that dimerize as a heterotetramer. And I'll explain how this fits together as we go on. But your effective machinery looks like this. So you're passing one segment of DNA through another. You've got a cup at the top called the N gate, which is made from this part of the polypeptide. And you've got a cup at the bottom, which is the C gate, which is made from the other part of the polypeptide. So you've got these two cups that face in different directions, and you've got a gate at the middle where the cups meet. And the first job of the gate is to bind the first segment of DNA, and this is coloured in green. And for reasons that I won't go into, that DNA is bent at the gate. Now, you imagine the rest of the genome is attached to this piece of DNA in green. You want to pass another part of it through it, and this is called the T segment for transfer segment, and this is in grey. And remember, you can't lose the ends of your DNA. You've got to keep them together. You've got to control this process. So the top cup, the N gate, via ATP hydrolysis, closes round the T segment and traps it. So this is stage 2, and you're ready to pass the DNA through the DNA. Upon hydrolysis of that ATP, you can cleave the first segment of DNA, but you hold on to it. Now you've got space, and you pass the T segment through it. Then your job is to make this unidirectional, because you're passing it through, and you want to trap it. So you religate the piece of DNA at the middle, and the bottom cup opens, and you let the T segment go. And then the cycle can begin again if necessary. So hopefully it should be readily apparent to you that when you mess about with this process, you're going to kill the cells. Or you're going to just generate DNA strand breaks, which are not good at all. Now, if you want to hold on to that first bit of DNA that you cut, you do it at the waist of the enzyme. and it is catalyzed by a conserved tyrosine, which is coloured in red here. And what you see is you've got this array of acidic residues, 1, 2, 3, 4 acidic residues. They bind to magnesium ions which neutralize the phosphodiester bond between two DNA bases. And you've got to separate the DNA, so your tyrosine comes in, gets activated by a base, and breaks the phosphodiester bond. And in doing so, you make a covalent linkage, so it's a covalent tyrosine phosphate linkage. So it's a neat mechanism in which you've clipped the DNA, but via the covalent interaction with the tyrosine, you've kept hold of it. So that strand isn't going to diffuse away. So this is how the DNA is both broken and retained. So if we go back, you have to break it and retain it, and so there's a physical linkage via the tyrosine to the end of that DNA strand. And because it's a dimer, you do so on the opposing strand of DNA also. I've written at the top, no structure of a full-length enzyme. This is no longer true. We have a low-resolution structure, which I'll describe in a few slides' time. But our best knowledge came from composite models. So, i.e., we would solve the structure of the top cup or the bottom cup in combination with the waste, and then we'd overlap on the waste, and we'd understand how the whole apparatus worked by piecing together two composite models. And there's an example of these two composite models. And when we're doing so, we can put a bit more detail on this apparatus. We can see that the N-terminal part of here has got the GHKL motif, and that's responsible for binding the DNA at the top. It's also responsible for binding ATP and performing a signal transduction event through this region in orange that talks to the waste of the enzyme. At the waste, you've got this domain in red, which binds that initial G piece of DNA that's bent. The bending and binding is helped by this shoulder region here, called the tower region. And then the bottom cup is formed of this winged helix domain in purple and this coiled coil in blue that closes at the bottom of the gate. There's an extra domain down here in certain family members. We don't know what that does. So here is the structure of that full-length enzyme. Note the rather appalling resolution at 4.4 angstroms. But at this resolution, we can still see what's kind of going on with the folds. The question is, do we see anything in the full-length structure that we didn't see in the composite models? And the answer is yes. We see a phenomenon called the domain swap. And a domain swap's something like this, right? If you've got a dimer with two domains, you can arrange them like that, running head to tail. But if they undergo a domain swap, you have the opportunity to do this, where the place of one takes the place of the other, and the domain swap has the net. So why did we see a domain swap in gyrase topoisomerase? It's really quite cool. You need the domain swap to drive the unidirectionality of the machinery. If you don't do that, then the DNA can move backwards, right? So what do I mean by that? So they've annoyingly coloured the two pieces of DNA opposite to what we did in the other slides. But anyway, you bind your first piece, you bend it, You then bind your second piece and you close the top end gate over it. Then, as you transfer that piece of DNA through the cleaved middle, the domain swap occurs. So you see in this figure that the end gate here was grey left, yellow right, and when you transfer the DNA, they see in the full length structure that it's yellow left, grey right. So what you've done is you've twisted the top of the enzyme 180 degrees. And the reason why you do that is the pivot closes off the top of the enzyme, and that's what gives you the unidirectionality. If you didn't twist the top of the enzyme, this cavity would still exist and the DNA could diffuse back up. So it's the domain swap like that that allows the reaction to occur in a unidirectional fashion. So here's a video of that. It's just a really cool mechanism, right? And then it opens up and let's go. How these machineries evolved to do this is quite remarkable, I think. Right, now, so the question is, I've taught you about gyrases, topoisomerases. How are they poisoned by fluoroquinolone antibiotics? So what was our first hint? So if we go way back to 2009, this is this paper by Lapanogov et al., where they propose that quinolones and fluoroquinolones act by providing stability to the cleaved complex, but not allowing it to be separated, so you don't pass the second piece of DNA through. So what did they see? They used the strep pneumoniae par C par E system. They've got the bottom gate and they've got the waist, and where the DNA is bent and cleaved, they see two molecules of fluoroquinolone. These are depicted in red. This is moxifloxacin, and if you go look at this PDB, you'll see the tyrosine has made a covalent linkage to the DNA, but capped where, just at the end of where you've made that breakage, and don't forget this is twofold symmetric, is your fluoroquinolone. And if you look at the detail of this, it kind of makes sense, right? You've got your DNA double helix, and you've got your two breakage points where you've tried to pull it apart, and that's one tyrosine up here, one down here. And at that physical position of breakage where you've got space, the fluoroquinolone stacks and fills the hole. And it's almost like, in a DNA double helix, I don't get to draw enough of these, where you've got the ladder, it's almost like when you've broken it, the fluoroquinolone sits and perpetuates that planar stacking. So you've broken the DNA, you've kept the structure intact by putting fluoroquinolones in where bases would normally be, and it's such a stable entity that you've blocked the ability to pull the strands apart and transfer the second segment of DNA. What's the caveats to this? 4 angstroms. Right, so they did a good job of describing the stability of the cleaved complex, but they did an absolutely appalling job of describing which way the fluoroquinolone entered the protein. So here's a description of that stacking. We'll just scoot past that for reasons of time. And then they must have kicked themselves when, a few years later, a much higher resolution complex was determined. This is from the pathogen Acinetobacter. And now they had such detail in their structure, they could show, the last one I showed you, they put the fluoroquinolone in backwards. So they modelled it wrong. So they had the right idea that it would block strand separation, but they had the wrong idea about the interactions the drug makes with the enzyme. And it's really neat now. So instead of going in this way, you see at 4 angstroms, you wouldn't be able to see the difference. At 2 angstroms, you can model this correctly. And they show that the acidic part, this OH part of the fluoroquinolone, makes an amazing interaction with the enzymes. This is glutamate, this is a serine. They bind a magnesium, and that binds the acidic part of the fluoroquinolone and allows it to sit upright between 2 bases. And this new higher resolution study has been validated because you absolutely need magnesium for fluoroquinolones to be active. If you remove magnesium, fluoroquinolones don't work. And the reason why is because magnesium potentiates an interaction between the acidic group of the drug and the acidic group of the enzyme. And look at the way stack, 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 drug, stack, the sort of DNA planarity is maintained by drug action. We'll have to continue next time in a bit. But how does resistance occur? So traditionally, you get really weak resistance to fluoroquinolones. So the first thing you do is you make an efflux pump, and you can pump them out, and you don't really become fully resistant, you become partially resistant. But what you can do is the region that interacts with the magnesium that interacts with the drug, is you can develop mutations. And this is known as the QRDR, which is the quinone resistance determining region. And you make mutations, for example, serine to leucine, or glutamate to lysine, that prevent the magnesium binding. And when you prevent that binding, you prevent the drug binding. And this gives you slightly stronger resistance. But in the 1990s, you have this phenomenon, which I hope is going to blow your minds in a minute, where you could have stronger resistance that was transferred on a plasmid. So instead of mutating your gyrase or your topoisomerase to remove this pocket, you acquired a gene that made you completely resistant, and no one understood why. So the genes that would arrive to make you fully resistant to fluoroquinolones we'll call this QNR family. And QNR family have a massive array of repeats in a row. So they're long proteins, but every 5 amino acids in turn follows a pattern. So the first amino acid in the repeat would be small. The second amino acid in the repeat would be capable of making a hydrogen bond. The third would always be hydrophobic. The fourth would always be polar. And the fifth was almost always glycine. And then it repeated again for several hundred amino acids. And this gave you resistance to fluoroquinolones. Anyone without looking at the handout want to postulate why? So nobody saw this coming, right? This is an amazing example. Nobody could have predicted this. And when they solved the structure of a QNR resistance protein, people got super excited. Right? So that repeat region makes up one side of a tube. So amino acids 1 to 5 has this repeat. Then you have another repeat, turns 90 degrees, 90 degrees, 90 degrees. So every 4 repeats, You go up one in this tube, and you make this structure here. And you can see that amino acid 1, you face solution, 2, you're small on the face, 3, you're hydrophobic to pack, 4, you're polar because you go out, and 5, you're glycine because you have to make a tight turn. And these repeats all line up on one another. And at the middle, there's a dimer interface that comes about from a helix that can pivot. So why did this give you resistance to DNA? Active drugs, fluoroquinolones. It's really cool. The protein mimics DNA. So here's the structure of the fluoroquinolone resistance protein. Here's the structure of DNA. You've even got grooves. And what the resistance protein does is it comes down, binds the middle gate of gyrase topoisomerase, and prevents the recruitment of the drug DNA complex. So it's really cool. Right, I will continue this next time.